So this is actually our institute. I am from Portugal, and we have been interested in two different topics, among other things, but that's the two examples that I decided to bring here, and to explain a little bit of what was the challenge of developing tools to study these two species. It is rice and cork oak. So they have nothing to do with each other, but they are both relevant for Portugal. Rice is very much consumed in Portugal. We are the bigger rice eaters in Europe, with about 15 kilos per capita a year. And we grow rice in three main riverbeds. And we cultivate about 29,000 hectares, and it is a really important crop. Regarding cork oak is also occupying a large part of the country in the forest area, and we have a huge production of cork, about 100,000 tons of production per year. So these are two examples. We started working in rice around the year 2000, and we started with cork oak in around 2007. And I will explain a little bit of the difficulties we went through to develop the tools, but how we are now. So the challenge of rice was because being such an important crop for Portugal, we had no breeding program going on anymore. So since the late 60s, we were importing seed from Italy, from the leftovers of the Italian breeding program, to have something to cultivate. And we noticed that the farmers were growing the Italian varieties because they produced more, but they had a small part of their land for the production of traditional Portuguese varieties for their own consumption because they tasted better. So we decided to target two of these varieties and try to bring them to a higher production level and to better characteristics for production. So we started a marker-assisted selection program for introducing the SME-dwarf gene because plants were pretty high, producing not enough, and to introduce blast resistance to Magna portorizae. So we did this for those two varieties using the marker-assisted selection. And interestingly, because we are in a campus where we have the agronomy station hosting us, actually, this raised the interest of the neighbors in doing something for rice, because if we were working in it, perhaps it deserved some attention. And it was interesting because in 2004, it was possible to raise the operational and technological rice center in what was already degrading the old rice center. But this time with the involvement of the producers and the industrials. And a few years later, this became a competence center that is now developing a national rice breeding program. So we were very happy with this. We had in collection over 1,200 different rice varieties from the old times in which we were developing breeding. So this was an excellent material to restart the program. We shifted our improved varieties to Cota Roche, that's the name we give it, and they followed the program, although not anymore using marker-assisted selection. So they used top crosses and mass selection to assess architecture of the plant and grain quality bench. And already in 2018, it was possible to start releasing varieties bred in this center, and they are already being sold and starting to be commercialized. Well, with this rice work, we started also thinking about some problems of the culture in Portugal. And the problems that we were told by the farmers that existed were low temperature in the north of Portugal. Remember, this was 20 years ago. And salinity in the center and south. In the north, they had solved the problem of salinity through stronger irrigation. But in the center and south, it was more difficult. So we started thinking about what to do with this. And in a previous study, we did a review in which we searched all KTLs described for rice and what could be related to, what could be carrying interesting genes related to these traits. And we became interested in OSCPK17 that segregates with 4 salt-related KTLs. But when we started studying it and characterizing its function, we found that it was more effective in cold stress response than in salt stress. Eventually also associated with sugar metabolism and activity of membrane channels, and this is being further studied by a colleague of mine. By the time when we thought about the problem of water use, nobody was interested. The breeders at the Cota Roche Center said that there's absolutely no interest because we use irrigation, so we have water and that's it. But 2 years ago, they shifted this mindset. And now they say, this is desperately needed. We have to shift the irrigation system from flooding to irrigated because this is not sustainable. So they are eager to have varieties that can cope with less water supply. So we started we were studying several things at the moment and characterizing several genes with analogies with other cereals and seeing how rice was reacting and what could be relevant in these kind of stresses. And we found some interactions that suggested that regulated genes were selectively targeted for degradation at certain points. And these led us, and sometimes these regulators, when they are overexpressed or knocked out, we see also some strong morphological changes, especially at root level, and roots are so important for all these environmental interactions. So we started thinking that perhaps we could look at what is able in the plant to have a fast response. And this fast response is usually achieved by proteins that are already in the cell and that are modified for being degraded, not needed anymore, or being activated and becoming active. So we started thinking about about this proteome degradation and looking a bit better into this. One of the researchers in the team, Tiago Lourenço, started thinking about targeting and investigating the E3 ubiquitin ligases that are key in this first response level and that are the most diverse group in this complex system because they define the target protein. So there are 2 main types of ubiquitin ligases, monomeric or multi-subunit. And this E-ring type, which is very abundant, in rice has about 425 different genes predicted. It's about the same in Arabidopsis with 1 more. So Tiago started investigating this using a candidate gene approach in which from these over 400 different ubiquitin ligases. He started searching in gene expression databases, characterizing the background and developmental stage in which these genes express, comparing with Arabidopsis information. And from this in silico study and bibliographic study, he selected 16 genes. These 16 genes are 14 cytoplasmic and 2 transmembrane, and he targeted some of them using overexpression and knockout to understand the relevance. So there has been a long track. I have to accelerate to characterize these genes and to develop gene editing tools. It is interesting because there are changes in the phenotype at the shoot and root level. So we have several other ongoing research topics in rice in an enlarged team with 2 colleagues that started as postdocs in my lab and now are co-working with us. But now I want to bring you to another, so the research in rice is going on pretty well. Now I want to bring you another subject, which is cork oak. I told you that cork oak is pretty interesting. We have a huge number of species of Quercus. Quercus suber is cork oak. And it has a very high interest in terms of sustainability and ecologic relevance because of the capture of CO2, being a barrier against fires, protecting the soil, creating employment and wealth, and having a huge ecological role. And cork is 100% recyclable and reusable. It is applied in a wide number of different applications, but one that you know better is cork stoppers. So in terms of economic, we have 1.2 billion euros in 2022, and the industry aims to reach 1.5 by 2030. And Portugal produces half of the world production and processes more than half of the world cork. So it's really a very important crop, species, not crop. But cork is harvested by hand using this tool. And so far, this is the most efficient way to remove the cork without damaging the tree. This is done during summer, once every 9 years. So the trees are labeled. And the first harvest is at 25 years of growth of the tree. Then the cork is stored, prepared. And there are many characteristics that define good or bad quality cork. The first one, that is called virgin, is only produced, it has the worst quality. And with after the third extraction, you really have the high quality cork. So this is an investment, if one wants to make it, for the grandchildren. And the main component of cork is a biopolyester, suberin. And the higher concentration of suberin, the best quality of cork. And there is an empirical knowledge that says that the cork quality is affected by the environment. So we wanted very much to study this, but we had no tools. It's such a long-living species, it's difficult to study. So we had a huge effort to try to mobilize researchers across the country and have the interest of the politicians and of the National Science Foundation in order to start with the transcriptome. And after many years of effort, we managed to establish a national consortium joining over 20 research teams from 11 different institutes and distributing work, and got the funding from the National Science Foundation to have 12 projects, 2 of bioinformatics, in order to do as much as possible the transcriptome of many different developmental stages, biotic and abiotic situations. The first paper of this joint effort came out in 2014, and then several others from the different teams came out later on. We also developed tools for epigenetic study while we still didn't have information from the genome, using a technique that is MSAP, so methylation-sensitive amplification polymorphism. It is a variation of the AFLP using restriction enzymes that are or not sensitive to methylation, and that allow it to identify a number of characteristics that appear to have an impact in cork formation. For instance, we characterized 3 different regions in collaboration with another team that was also interested in the subject. We found that populations differed widely in cork quality, but only a little bit in epigenetic differentiation. We found different methylation profiles between leaves and cork. We found really interesting variation, epigenetic variability between virgin cork, the first one, and Amadia cork, which is the one that is already high quality after the third one. And 4 markers that have significant associations with porosity, the pores that form in the cork that you want to reduce to have by high quality, and so-called nail. Nail is wood inclusions, and that happens when the meristem that forms this cork layer, the phellem, fails to be continuous, and then there is wood in between, and this is of course bad. So later on, we managed to have, after many different efforts, and I will skip the details, to have a genome sequencing initiative. And we could have it funded with the European funds for regional development in Alentejo, in the south of Portugal. And this allowed developing the first draft genome sequence of cork oak in 2018, and just 2 months ago we managed to publish the improved reference genome and first organelle genomes of cork oak. So only to target the tree to sequence the genome was a challenge, because it hybridizes a lot. So we wanted to minimize this hybridization, because some of the hybrids, they don't have good quality cork. So we had to analyze different trees that were reported to have good quality cork and minimize the level of heterozygosity. So, and we selected, this was the candidate that we have used. So I told you that we have indeed an impact of climate on development of cork. And here you can see that in 2005, when we had a serious period of drought, the cork development was shorter. So I have to skip a lot of details, but I will say that we wanted to study the cork formation and the impact of environmental cues on this formation, and we had to target something that we could use fast. So we started analyzing Arabidopsis for the formation of for the suberization of the periderm that happens at the top level of the root, and then transfer to cork oak roots to transfer some of the knowledge. In Arabidopsis, of course, it was easy to use the technology of TrapSec in order to have the specific transcriptome of the suberizing cells in this layer. We correlated with chemical composition, and we did a precise morphological study in order to identify when the cells start to activate genes responsive, involved in the biosynthetic pathway. Then we shifted this study to cork oak, and also had to go through the morphological study and compare the secondary development zone with the primary development zone in order to have this difference, to identify the genes of interest, deep characterization of the process, And then we shifted to stems to understand what goes on at stem level. But for this, we needed plants already with at least 1 year and a half of growth. And we submitted them to extended drought period over 6 months. And the interesting thing I would like to show you is that this, so it is possible to remove the cork part, the phellem, separate it from the inner bark. These are the fingertips, and isolate different tissues for doing the transcriptomics. And we found a number of different genes. This is still not published. This work is still not published, but we found genes that are interested and have been described as being expressing at this phylogen level. But also, interestingly, I'll shift, I'm finishing. We found that there is a correlation with the increase of photosynthesis in extended drought period. And this shows that there is an improvement of the photosynthesis in this extended period. So, to finish, I'm sorry, I'm skipping a lot of details. This effort, it was good because we could establish a lot of tools that are available for the whole country. And now we have not only genomic tools, but also bioinformatic tools and collaborations and integration in European infrastructure of bioinformatics, Elixir, and this allowed a strong progress. So I want to acknowledge my team, and especially Tiago, who is leading now the rice work. Pedro Barros is leading the cork oak work. Nelson and Isabel were former members and are now PIs. They are developing the rest of the work in rice, and some former students. And we have many collaborators. I could not name them all. And thank you for your attention.